polymerase chain reaction process was created by Carey Mullis in 1990. The polymerase chain reaction is normally called PCR. There are 6 main types of PCR. They are real-time PCR, nested PCR, multiplex PCR, quantitative PCR, qualitative PCR, and arbitrary PCR. The main ones I am going to talk about are qualitative PCR and real-time PCR. PCR was a revolutionary process. It has been the building block that all of today's microbiology has been built off of. PCR can take the tiniest amount of DNA and make mass amounts. It is truly an amazing process. It takes a tiny microscopic piece and makes it so we have the ability to test and view it. It can also detect if there is DNA in a sample. This is known as qualitative PCR. All PCR uses the same process to recreate more DNA. The process all starts with the same ingredients: template DNA, primers, nucleotides, and DNA polymerase. All of these ingredients are combined and put together in a thermocycler. Within the thermocycler, there's a square receptacle that controls temperature. This container raises and lowers temperatures to control the temperatures of the test tubes containing ingredients that were put in the square container. The first temperature breaks apart the DNA. The breaking of DNA is known as denaturation. Then it cools back down so that the specific primers can attach to the separated DNA pieces. These specific primers are designed according to the target DNA sequence in their experiment by the individual scientist. This process is known as annealing. To anneal the primers in the target DNA sequence, they must have a complementary sequence. Then it is brought to an even hotter temperature. This is when DNA polymerase that was extracted from the bacterium Thermus aquaticus starts to do its job. This DNA polymerase enzyme is able to do its job at much higher temperatures because it comes from the hot springs. The reason this is so revolutionary is because the DNA polymerase is no longer destroyed by the heat required to separate DNA pieces. During this third temperature, the DNA polymerase takes the nucleotides and extends the primers to create a new DNA strand. Each time the thermocycler goes through the cycle containing these 3 temperatures, it doubles the amount of DNA. It went from 1 piece of DNA to 2. The differences between the PCR processes are the way they are collected, compared, and measured. Most of the time, scientists use agarose gel electrophoresis to view and be able to compare DNA strands. When they use the process of agarose gel electrophoresis to view the results, they have 2 options: add ethidium bromide or some type of fluorescent dye. Qualitative PCR uses this method to view its results. Qualitative is mainly used to discover if DNA is present in a sample. This is what qualitative would look like. Real-time PCR is a much more detailed way of getting information. It allows for a more detailed report giving exact measurements of DNA. It is able to tell the exact amount of DNA the scientists started with and ended with. Polymerase chain reactions are such a revolutionary discovery. They made so many things possible just by making something so tiny such as DNA, into large amounts. PCR is used in so many different fields, such as research labs, genetics, forensics, medical diagnosis, and so much more.